Hello friends, today's topic of discussion is penicillin fermentation. As we all know, penicillin is a type of an antibiotic. Antibiotic is an organic compound produced by one microorganism that at greater dilutions inhibits the growth of other group of microorganisms. Antibiotics are available in various forms like in an ointment, cream or an injection form. So according to our requirement, we need to utilize specific form of antibiotic. Antibiotics are produced primarily by bacteria, streptomyces, nocardia and fungi. There are a number of ways available for the classification of antibiotics. But on the basis of mode of action, antibiotics are divided into number of groups like antibiotics affecting cell wall, antibiotics damaging cell membrane, antibiotics interfering with protein synthesis, antibiotics inhibiting nucleic acid synthesis, and antibiotics blocking cell metabolism. Penicillin. Penicillin is first discovered natural antibiotic. It was obtained from multicellular fungi, that is Penicillium molds. Penicillin is a group of compounds having common basic nucleus which is termed as beta-lactam ring, that is 6-amino-penicillinic acid. There are two types of penicillin, natural penicillin and semi-synthetic penicillin. Now you can see the structure of beta-lactam ring of penicillin where number of side chains remain present. Now whenever we'll try to remove side chains which remain present with natural penicillin by any other side chain, it is termed as semi-synthetic penicillin. In 1928, Sir Alexander Fleming first observed antibiotic properties and therapeutic value of penicillin. In an experiment, he observed that airborne contaminant, later shown to be Penicillium notatum, inhibited the growth of bacterial culture, that is S. aureus. He called this material penicillin after the mold that had produced it but was unable to isolate it. By that, the progress of the first modern era antibiotic, that is penicillin, gets started. In 1940, Howard Florey and Dr. Ernst B. Chain initiated extracting penicillin into a purified and powerful antibiotic. By the end of the war, that is in late 1943, mass production of the penicillin had started by many drug manufacturing companies, and because of this contribution in 1945, Sir Alexander Fleming, Florey and Chain were awarded the Nobel Prize in the field of physiology and medicine. Now mode of action of penicillin. Penicillin mainly inhibits enzymes involved in the cross-linking step of peptidoglycan synthesis in the cell wall. As we all know, peptidoglycan is the major component of bacterial cell wall, but concentration of peptidoglycan is higher in gram-positive cell wall as compared to gram-negative cell wall. So, penicillin is effective against many gram-positive bacteria, nocardia and actinomycetes, but not against most gram-negative bacteria except at higher dosage level. The World War II had brought a demand for penicillin on a large scale for the treatment of burns and wounds. Penicillin fermentation. Fermentation is the process used for the large-scale production of an antibiotic. Out of various species of the fungus Penicillium, mainly two species, that is Penicillium notatum and Penicillium chrysogenum, are widely utilized. Even from these two, Penicillium chrysogenum is a high-yielding strain whenever mutated by X-rays. These are some pictures of Penicillium, where septate mycelial structure having conidiospores. Whenever we want to start large-scale production of penicillin by fermentation, our first task will be inoculum preparation. To develop a pure inoculum in an adequate amount, the fast-growing phase for the large fermenter following steps are necessary. A starter culture available in cold stored form is transferred to an agar-containing plate to allow growth. After getting growth on solid media, one or two growth stages should be allowed in shaken flask, which can transfer to seed tank for further growth. After about 24 to 28 hours, the content of the seed tank is transferred to the primary fermentation tank. The main fermentation tank is a larger version of the seed tank which contains same growth media and also provides proper growth promoting environment. During this process, they excrete huge amounts of the desired antibiotic. Beside inoculum preparation, other bioparameters should be maintained, like pH should be near about 6.5, temperature in between 26 degrees centigrade to 28 degrees centigrade, continuous aeration is provided by steam or sterilized air, agitation is provided by baffles. Raw materials contains all the necessary elements like carbon source, nitrogen source, mineral source, precursors and antiform agents if necessary. Raw materials are required for the proliferation of microorganisms. Following 3 points should be kept in mind whenever we are going to choose any raw material. First, an abundant growth of mycelium. Second, it should allow maximum accumulation of our product, that is penicillin, and it should make downstream process, that is extraction and purification of product, easy. Carbon source. As a carbon source, lactose acts a very satisfactory carbon compound whenever used in a concentration of 6%. Other carbohydrates like glucose and sucrose may be used, but it has to provide with slow feeding rate. Complex as well as cheap sources like molasses or soy milk can also be used, which are made up of lactose and glucose sugars. Penicillin is a secondary metabolite like all other antibiotics, so it is only produced in the stationary phase. So in figure you can able to understand that as cells will grow and once they will reach to stationary phase, penicillin production gets started and lactose degradation gets started. So by the utilization of lactose, penicillin production is there. Nitrogen and mineral source. Ammonium salts such as ammonium sulfate, ammonium acetate, lactate or ammonia gas are used as nitrogen source. Sometimes complex source, that is Corn-Stip liquor, may also be used. Additionally, some minerals like phosphorus, sulfur, magnesium, zinc, iron and copper are added in the form of water-soluble salts. The most important naturally occurring penicillin is penicillin G. But depending upon the precursors added, the type of penicillin going to produce, that can be changed. For example, if we are going to add phenylacetic acid as a precursor, penicillin G is being produced. But instead of that, if we use hydroxyphenylacetic acid, it will allow production of penicillin X. And by addition of phenoxyacetic acid, we can produce penicillin V on large scale. Last step of penicillin fermentation is the recovery. The recovery of penicillin is carried out in 3 successive stages: removal of mycelium, countercurrent solvent extraction of penicillin, and treatment of crude extract. In downstream processing, there is no need to break open the fungal cells as penicillin is extracellular product. As penicillin is a therapeutic medical drug, it needs to be very pure. After purification, it is precipitated as potassium salt to separate it from other substances in the medium. The resulting penicillin can be used to make a variety of other penicillins like penicillin V, penicillin O and ampicillin. So this is the chart which will give you idea about penicillin fermentation, that our first necessity is formation of production medium. At the same time, we need to prepare inoculum from starter culture, that is Penicillium chrysogenum. After that, we need to prepare main fermenter where we are inoculating our inoculum medium to the production medium. Then 6 to 7 days in batch fermenter we need to allow incubation. Then we need to start downstream processing. So first, with the help of rotatory filter, we need to remove mycelial structure. Then by countercurrent solvent extraction, by using different solvent, We need to allow purification of penicillin, and then the final penicillin salt, either sodium or potassium salt of penicillin, we need to treat to produce purified penicillin. Then after rotatory filter treatment, the mycelial structure which we will get at the end, that we can utilize for animal feed. After purification, penicillin will be packaged either in the form of cream or tablet or injection. So in different form we can allow use of penicillin. Thank you.